Hi everyone, my name is Sabrina Afifah Binti A.Karim and I am from MS4. Today, I would like to discuss about transcription. Transcription occurs in nucleus. During transcription, mRNA is synthesized using a specific segment of DNA as a template. The segment of DNA is known as a gene. This is a gene. A single gene is a segment of the DNA that codes for the synthesis of a specific protein. A gene consists of a promoter site. This is promoter site. Followed by the mRNA coding sequence for the synthesis of a protein. And then a terminator site. This is terminator site. The RNA coding sequence is a sequence of base pairs that contains coding information for the polypeptide chain specified by the gene. The terminator site is a sequence of base pair that specify the end of mRNA transcript. The synthesis of DNA during DNA replication, the synthesis of mRNA also occurs in 5' to 3'. But the enzyme involved in the process RNA polymerase.
Hello everyone, my name is Teh Di Haen and I'm from MS4. Today I would like to talk about transcription. There are 3 stages in the transcription of DNA to RNA, that is, initiation, elongation, and termination. For now, I would like to talk about initiation. In prokaryotes, initiation of transcription occurs with the specific recognition of the promoter sequence by RNA polymerase, whereas the initiation in eukaryotes is more complex, whereby RNA polymerase recognizes a complex of proteins that help with the binding of the enzyme to the promoter sequence. Firstly, in transcription, the RNA polymerase binds to the promoter site of DNA. It moves along the DNA in the direction of transition and unwinds the double helix DNA by breaking the hydrogen bond between the nitrogenous bases of the polynucleotide. This causes both DNA strands to be separated. After separating, only one DNA strand is used as a template for transcription. Now, I would like to demonstrate on the process of transcription using this model that we have just made. Firstly, the RNA polymerase binds to the promoter site of the DNA. Next, it moves along the DNA in the direction of the transcription and it unwinds the double helix DNA. The hydrogen bond between the nitrogenous bases are broken, and this causes both the strands to be separated. Therefore, only one DNA strand is used as the template for transcription.
My name is Nurul Syafrina Binti Ngateman. I'm from MS4. I will introduce step 2, which is elongation. In elongation, RNA polymerase add nucleotide in direction of 5' to 3' end. Then, RNA polymerase add a free ribonucleotide which are complementary to the bases on the DNA template strand. Cytosine pairs with guanine, uracil pair with adenine. DNA-RNA hybrid is temporarily formed.
Hello, my name is Siti Rahmah binti Sanir from MS4. Today, I will explain more about termination process in transcription. Elongation proceeds until RNA polymerase reach termination site. terminator sequence gives signal to RNA polymerase to stop adenucleotide to mRNA strand. RNA polymerase will detach from the DNA strand. mRNA molecule is released from the DNA template. Double-stranded DNA will rejoin to form double helix again. In prokaryote, mRNA doesn't need any modification, while in eukaryote, further modification is needed. Thus, after transcription, its mRNA is called as pre-mRNA and will be used for translation process.
Hi everyone, my name is Teh Di Haen and I'm from MS4. Now I would like to talk about the process of RNA splicing. Transcription in eukaryotic cells differs from prokaryotic cells in that the RNA transcript is a pre-mRNA molecule which requires modification before they can leave the nucleus. This is because many eukaryotic genes contain known coding segments called introns that break out the amino acid coding sequence into segments known as exons. Exons are coding segments in genes. During transcription, RNA polymerase will transcribe both exons and introns from the DNA, but the introns will not be translated. The introns are removed from the pre-mRNA strand and the exons are then joined together to form a mature mRNA molecule with continuous coding sequence. This process, which is known as RNA processing or RNA splicing, requires the aid of many enzymes. One of the examples is spliceosome enzyme. Now, I'd like to show an example of the RNA splicing process. In this case, coding sequences, exons, and non-coding sequences, introns, are found along a mRNA strand in eukaryotes. Coding sequences are not continuous, as introns are found between the coding regions, the exons. Pre-mRNA contain exon and intron regions. Process of removing the intron and recombining all the coding exons regions are carried out by the RNA splicing in the nucleus. In RNA splicing process, the introns are cut off from the pro-mRNA by the enzyme spliceosome. Spliceosome binds to specific nucleotide sequence along an intron to cut it out and release it from the pre-mRNA strand. After the introns are removed, the exon that flank the intron are joined together. Therefore, a functional mRNA is produced and the mRNA strand moves from the nucleus into the cytoplasm of the ribosome to be translated so that the process of protein synthesis can be carried out.